Very good morning, students. Today we are going to learn about very important metabolic pathway in microbial physiology and metabolism. That is fatty acid biosynthesis. So this fatty acid biosynthesis is a very complex pathway, and this is controlled by a set of enzymes. The first one is fatty acid synthase. So this is called as mentioned here as an enzyme. Usually this enzyme having a cofactor, that is a biotin. Usually it is a cofactor, and this cofactor is involved in the it is a carrier of carbon dioxide. So that is why this enzyme biotin complex is involved in the first reaction, and this first reaction is here, and in this first reaction an enzyme plus biotin, okay, this enzyme B is there, plus ATP plus HCO3. Okay, that is bicarbonate ion. So acetyl CoA carboxylase. So no need to mention okay about this one. This is a name of the gene. Okay, this is not needed here. This may be in the advanced level. And acetyl CoA carboxylase is enzyme, and this carboxylation. This is carboxylation. So this carbon dioxide is going to be attached with biotin. Usually the biotin will receive only the carbon dioxide. Without biotin, the carbon dioxide will not be received. So you have to be very clear about that one. That is why the biotin enzyme complex receives carbon dioxide, and this reaction utilizes energy. That is why ATP is converted into ADP plus PI. Simple, that's all. So this CO3 donates CO2, and enzyme plus B is here. I hope okay, this is very simple reaction. And next reaction is acetyl CoA carboxylase. Carboxylation. Acetyl CoA carboxylase. The same enzyme. Okay, next enzyme, next reaction. The acetyl coenzyme A is combined with this one. That is this enzyme biotin with the carbon dioxide complex reacts with acetyl coenzyme A by the same enzyme acetyl CoA carboxylase. For the both reactions, the same enzyme, and this will form, for example, the underlined one is the same molecule CH3COSCOA, that is acetyl coenzyme E. Just you can imagine this CO2 is attached to here. This is COOH is a CO2. So this CO2 of enzyme plus biotin complex donates carbon dioxide to this acetyl coenzyme A. Now this acetyl coenzyme A, the underlined one is here, and the enzyme biotin complex is liberated, and the CO2 is attached with here. Next one is next reaction is malonyl CoA ACP transacetylase. Sorry, malonyl CoA malonyl CoA ACP transacetylase, and this acylation acyl moiety is transferred, okay, from one component to another component. Okay, this is ACP. ACP means this acyl carrier protein. I already mentioned here. ACP means acyl carrier protein, and this is acetyl coenzyme ACP acetylase, and all the three-letter code designates each and every protein. Okay, this is regarding this one, the gene name and all. Okay, no need to remember that one. So we have to focus only these type of reactions. So this reaction is very important, malonyl coenzyme A, because the product is malonyl coenzyme A. This is the malonyl coenzyme A, and so this malonyl coenzyme A and ACP transacetylase. Okay, that is ACP means acyl carrier protein of the enzyme. Okay, the enzyme having acyl carrier protein region. So that region having the active, that is functional group is SH group. So this SH group reacts with, so this component, this is substrate, and after the reaction this will form ACP attached with here. That is in the place of coenzyme A, ACP is going to be attached. Just you can have a look. Okay, this is coenzyme A is liberated as a CoA-SH. In the place of coenzyme S, CoA, and CoACP is going to be attached. This CoACP is going to be attached. That's all. So that is the function. ACP is going to be attached in the place of coenzyme A, and coenzyme A is liberated now. Now this is the active one. So A, without acyl carrier protein, this fatty acid synthesis will not run. The major component of fatty acid synthesis is acyl carrier protein of fatty acid synthase. So this ACP attached with acyl moiety is the active one, and now this will undergo a sequence of biochemical reactions to form an elongated chain of fatty acid. So this is very simple. Just we can have a look. So this is the malonyl coenzyme with ACP. That is why this malonyl ACP, okay, this one, because coenzyme is liberated now. So malonyl ACP, so this malonyl ACP reacts with another coenzyme A attached with ACP. Okay, that is, so this is coenzyme A. Okay, this is coenzyme A, and coenzyme A is liberated. In the place of coenzyme A, ACP is going to be attached, and the name of the enzyme is TA. TA means transacetylase. Transacetylase, okay, this one. And ACP moiety is going to be attached with coenzyme A portion. Okay, this is acetyl coenzyme A, that is a product of pyruvate dehydrogenase complex, and this one is going to be reacted with ACP-SH, and this ACP is going to be attached with here. That is why this SACP is formed. And this SACP is formed, and this one going to react with here, okay, this component. So CH3CO moiety is going to be attached, okay, here, and SACP is liberated here, okay, and this COO moiety, this COOH is liberated as a carbon dioxide. So just imagine, okay, this is malonyl ACP. So this malonyl ACP, the CO2 is liberated, this COOH is liberated. That is why I made a point here. CO2 is liberated here, and the remaining ACP is liberated, and ACP is liberated in the form of ACP-SH. And this CH2O, the CH2O is going to be attached with, so this CH3CO. So CH3CO and CH2O, again SACP is there. So this SACP is going to be attached with the CH2O, and now CH3CO, CH2CO, SACP. So CH2CO SACP is already available, and CH3CO moiety is going to be attached. So that is why it is called as beta ketoacyl ACP synthesis 1, 2, and 3. So there are three genes, of set of genes are there, and maybe it depends on the microorganism, and it will act on the fatty acid formation. And the next one is beta ketoacyl ACP reductase. So this is reduction. Reduction means it requires NADPH. I already told, maybe explained at the time of taking HMP shunt pathway. The major function of HMP shunt pathway is production of ribose and the production of NADPH. So this is the key role. That is, NADPH is involved in the fatty acid synthesis. So NADPH plus H plus, okay, involved in this reduction reaction, and just you can have a look, okay, what is going to happen. So this CH3C is there, and this C and O. C is attached with hydrogen and O is attached with hydrogen. That means CH and OH is there. You can see the red color underlined one, that is the new two hydrogens are coming from NADPH plus H. That is H and H, another 2H. Two hydrogens are attached. So next one is beta hydroxyacyl ACP dehydratase. Dehydratase means the removal of water. So that is why I made, okay, this one clear. That is the OH of this portion and one hydrogen from the CH2. So that means H2O is liberated, and remaining fragment is CH3, CH, and double bond, because carbon is having 4 valency. Okay, this is a double bond. CH, CO, SACP. CH, CH, CO, SACP. And water molecule is liberated. And again, enoyl ACP reductase. Again, reduction. Reduction means here also NADPH and here also NADPH. And the CH is converted to CH2 and CH again, okay, this CH, one hydrogen is added, that is why it is made to CH2. And in this diagram, okay, CH2 is missed, okay, that is why I wrote here CH2 again, another CH2 is going to be formed again, COSACP, remaining fragment is COSACP, COSACP, NADP is liberated. And this one again going to join here as a elongation process. And this malonyl ACP again reacts with this one, and maybe this fragment, okay, this except SACP, CH3, CH2, CH2CO is going to be attached with the CH2CO. So each and every time, okay, two carbons are going to be attached, and ultimately a lengthy polymer is going to be formed. Polymer means, okay, the fatty acid is going to be formed. For example, for the formation of C18, C18 means, okay, each and every time two carbon has to be attached. So each and every two carbon is going to be attached, and finally C18 will be formed. C18, C20, okay, likewise. I think so. This is a very simple one, and maybe if you're understanding the concept, it's very easy to understand. Okay, and this is very important for the examination point of view. And this overall reaction, this is the lengthy polysaccharide. Sorry, that is fatty acid. So this fatty acid is produced only by fatty acid synthase molecule. So with this, we can conclude the fatty acid synthesis. Next, we can go to the phospholipids. The phospholipids are simply called as phosphorylated lipids, a lipid containing phosphates. Okay, that is why it is called as phosphorylated lipids. And so these are all very important components of the biochemical pathways and biochemical machinery of the cell. And the phospholipids are very important in that is the 3D shape, to maintain the 3D shape of the membrane. Okay, there is a lipid bilayer membrane, and as a component of a bacterial cell, and lipid bilayer plays a major role to diffuse or to transport the metabolites, okay, from outside to inner and inner to outside. So this one is play a very important role. Without phospholipids, there is no life also, because this is a very important one. Just you can have a look. We have so many different types of phospholipids: phosphatidyl serine, phosphatidyl ethanolamine, and phosphatidyl glycerol, and cardiolipin. And the basic fragment is phosphatidic acid. Just you can have a look. This is phosphatidic acid, and here in this place, if in the place of X, okay, if it is hydrogen, that is phosphatidic acid. So this is phosphoglyceride. Okay, it means the phospho and the glycerol moiety. I think many of you know that. Okay, what is the meaning of glycerol? Glycerol means CH2OH, CHOH, and the CH2OH. Okay, this is a glycerol moiety, and to the glycerol, and one R1 means the fatty acid is attached, and R2 is another fatty acid. R1 is first fatty acid to the carbon 1, and second fatty acid to the carbon 2. And here a phosphate is going to be attached in the form of phosphoric acid, okay, is to the third carbon, and to the phosphate an X molecule. X molecule is the reactive molecule is going to be attached. So this, usually the X will be the organic molecule. You can see here the serine, ethanolamine, glycerol. So mostly all the the X molecules are more polar. So that is why the schematically it is represented as, you can see here, this is the schematic representation of fatty acid. Usually you know the drawing of the membrane. The membrane, okay, you will draw, okay, the one layer is be a head and two tails. So this head represents including this and the phosphate, okay, because this is also highly soluble portion. And this is the fragment, and usually this is a base here, and these two chains are represented as R1 and R2. That's all. So basically, okay, this is a classification of phospholipids. One is phosphatidyl serine, phosphatidyl ethanolamine, phosphatidyl glycerol, and the phosphatidyl choline is also there, and we can see it later. And so X is here. This is phosphatidyl serine. Okay, this is serine, and ethanolamine. Okay, this is glycerol. Okay, and this is cardiolipin. So first one is in the phospholipid biosynthesis. We can go to the biosynthesis first, and we need to get, okay, all the phosphatidyl serine, phosphatidyl glycerol, and phosphatidyl ethanolamine, and phosphatidyl choline. Just you can have a look. Okay, this is phosphatidyl choline, and phosphatidyl ethanolamine, and phosphatidyl serine. And here, if you are coming to the second pathway, okay, that is phosphatidyl glycerol, phosphoglycerol phosphate, and phosphatidyl glycerol, and finally a cardiolipin. So the base molecule is this one. So this is the base, that is CDP diglyceride. What is CDP? Cytidine diphosphate diglyceride. Cytidine, COAT is cytidine, okay, and diphosphate, and this is glyceride. Glyceride means a glycerol with the two fatty acid chains. So that is the starting material for the synthesis of phospholipid, and this will undergo the first reaction, okay, that is called a CDP diacyl glyceride and serine O phosphatidyl transferase. So it's simply phosphatidyl transferase reaction. So phosphatidyl glycerol moiety is there. Okay, this phosphatidyl the glycerol moiety is going to be transferred, okay, to the another molecule. So here the serine is converted to CMP, that is CDP, and one phosphate is liberated, and the one phosphate is this is CDP, cytidine diphosphate, and one phosphate is already in the hold position. Okay, that means so this one CYT plus phosphate, that is cytidine monophosphate, so this is liberated, and these two, I'll take a pen and explain to you, that is better. And these two moieties are going to be removed as CMP, and the remaining portion, okay, this one attached with the serine, okay, this is a serine molecule, and this is the serine is coming inside, and a phosphate, this is a phosphate, and this is glyceride, diglyceride. So these two are these are the same, and this and the fragment is same. So this and this is same. Okay, the serine is going to be attached, and at the time of serine attachment, the CMP is removed. Now this is called as phosphatidyl serine. I'll clear this one. Okay, now I am going to take the pen again. Yes, next one is phosphatidylserine to phosphatidylethanolamine. So now the second reaction means phosphatidylserine decarboxylase. Decarboxylase means the removal of carbon dioxide. So the carbon dioxide is going to be liberated from here. So that is from the serine molecule, this COOH moiety is liberated as carbon dioxide, and the remaining portion is same. Okay, this is the remaining portion, and this remaining portion is called as phosphatidylethanolamine. So that's all. And next one is phosphatidylethanolamine to phosphatidylcholine. Choline means choline is an organic molecule, and SAM, S-adenosyl methionine, and S-adenosyl hydrogen. Okay, these two are S-methionine and S-adenosyl homocysteine. Okay, this homocysteine is going to be formed from S-adenosyl methionine, and the choline is going to be transferred, okay, to phosphatidylethanolamine, and so this is a very important pathway, okay, for the formation of phosphatidylcholine. Phosphatidylcholine is a very important component in the membrane, and this is a very important one for the fluidity of the membrane, the 3D shape and the flip-flop movement of all the lipid bilayer components. The fourth reaction is, so this glycerol 3 phosphate is going to be reacting with the CDP diacylglycerol, and so this one, after the reaction, okay, again this CMP is going to be liberated. Okay, this is very simple, and so this CDP diacyl diglyceride and glycerol 3 phosphate is going to be attached. So by the removal of this moiety, so CMP is going to be liberated, and so this is CH2OR and CHOR. So this is there, CH2OR, CHOR, and this is the phosphate, and this is the phosphate, and remaining glycerol 3 phosphate. Glycerol 3 phosphate means CH2, CH2, and with the phosphate. So this is glyceraldehyde. So this is glycerol 3 phosphate. So this glycerol 3 phosphate is going to be attached by the removal of CMP, and the phosphatidyl glycerol phosphate is there, and the phosphate is going to be liberated. Okay, phosphatidyl glycerol going to be formed by the removal of phosphate by the fifth enzyme. The fourth enzyme is CDP diacylglyceride glycerol 3 phosphate 3 phosphatidyl transferase. Phosphatidyl transferase is the name of the enzyme, and here the fifth enzyme is phosphatidyl glycerol phosphatase. Phosphatidyl glycerol phosphatase. Phosphatase means the removal of phosphate. So this phosphate is going to be liberated. This phosphate is going to be liberated, and as a PI. So this is the PI going to be liberated, and remaining fragment is, just you can have a look. Okay, this CH2 CH. So this CH2 CH CH2O phosphate and remaining fragment. Okay, here By the removal of phosphate, this O is going to be attached with the hydrogen. So that is why CH2OH will be there in the last. Okay, right. So next reaction is the incoming CDP diglyceride. This is not in your syllabus, but anyway, I will show it to you. CDP diglyceride is again reacting with this one, phosphatidyl phosphate, and CMP is liberated, okay, from CDP. And if the CMP is liberated, okay, this portion is going to be attached with this one. Yeah, sorry, this one. Yeah, this portion is going to be attached with this one. So that means, so to the phosphate we have to attach. So this one is going to be attached with the phosphate, and you can see here this is the one going to be attached with the phosphate. Okay, and the CMP is liberated, and so this is the remaining fragment. It's called as cardiolipin. So cardiolipin also a very important one, and not in the microorganisms. Okay, maybe very important for the eukaryotic system. So with this, we can conclude the phospholipid biosynthesis. Next, we can go to the biosynthesis of aspartate, pyruvate, and histidine and serine amino acid families. Amino acid biosynthesis. So this we can have it in the next session. And with this, we can conclude all the phospholipid biosynthesis and fatty acid biosynthesis.